Good afternoon, everyone. Myself, Gaurav Gupta. I am a lecturer in Life Science Department. Today, we are going to discuss about a drug targets. Under this one, today first we'll discuss about a receptors. As we know about a receptors, these receptors are a protein macromolecules which are present on a cell membrane or inside a cell in a cytosol or inside a nucleus. These are the receptors where the chemical messengers or any type of drugs they are binding and they are producing any type of cellular response inside a cell. That's called a receptor. So today we'll discuss about all these things, how they are functioning, what is the structure of receptors, how the drugs or the chemical messengers they are bounding to the receptors. That we'll discuss in this lecture. So the basic learning outcome of this lecture is, first we'll discuss about a structure and function of receptors, then about a messenger bindings, then overall process of receptor and messenger interaction, and laterly in last about a superfamilies, different classification of receptors we'll discuss. Under structure and function of receptors, you can see here in a figure there is a cell, and you can see on the above a cell there is a nerve. Between a nerve and cell, this small gap is there. That gap is called a synapses. That gap is called a synapses. So in a nerve, you can see here the signal arrow is showing towards the downside. So whenever the nerve impulses or the currents they are passing towards the end of nerves, these nerves they are releasing a chemical. Releasing a chemical. This chemical is called a messenger or a ligands. You can see here on the purple color they are releasing a chemical. This chemical is called a messenger or ligands. So these messengers they are binding with the cell on a cell surface receptors. They are binding with the cell surface receptors and they are producing any type of cellular responses that we can see in this next figure. You can see here after binding they are producing different type of responses. These responses like any changes in a protein structure or a new protein synthesis or any type of responses it may be. So you say structure and function of receptors. Here, so this receptor is working like a letterboxes which are receiving a letter. So here the letter indicating a chemical messengers which are attaching with the receptors and producing any type of responses inside a cell. So you can see here about the receptors. Receptors are a globular proteins acting as a cell's letterboxes. So the receptors is working as a letterboxes on a cell. They are located in a cell membrane. So along with their location on a cell membrane, they are also present in a cytosol, means inside a cell or inside a nucleus. What is the basic function of receptors? They are receiving a chemical messengers. They are receiving a messages from a different cells and they are passing that messages inside a cell in different type of reactions that we'll discuss in the next slides. Next is transmit a message into a cell leading to a cellular effect. So after receiving a messages from a chemical messengers, they are transmitting that message inside a cell in form of different type of reactions that we'll discuss later in the slides. Next point is different receptors specific for different chemical messengers. So this is a very important point. For each and every type of chemical messengers there is a specific receptors are there. So for each and every receptors they are accepting a specific type of chemical. Each cell has a range of receptors in a cell membrane making it responsive to different chemical messengers. So in each and every type of cells they having a range of receptors where the chemical messengers are binding and producing different type of reactions inside a cell. So this is a major functions and the structure of receptor. So now let's discuss about a chemical messengers as we discussed in our last slide about a chemical messengers. What is chemical messengers? Chemical messengers are a messengers which are passing informations to a other cell. So here these chemical messengers are two types. First, neurotransmitters. Neurotransmitters are a chemicals which are releasing from a nerve. That's called a neurotransmitters. As the name indicate, neuro denoting a nerves. So when the chemicals are releasing from a nerve, it's called a neurotransmitters. Hormones, a chemicals which are releasing from a cells or a glands and binding on a cells, that's called a hormone. So it's very simple. There's a two type of chemicals messengers. First, neurotransmitters, a chemical messengers which are releasing from a nerve, it's called a neurotransmitters. Second chemical messenger is hormones, chemicals which are releasing from a glands or from a cells, is called a hormones. So there's a two type of chemical messengers. Next, now you can see here from a nerve at the terminal point, chemical is releasing. This chemical is called a neurotransmitters, binding on a cells, producing any type of responses. Next, hormones from a cells or from a glands, the chemicals are releasing and transferring or traveling through the blood supply and reaches to a particular target cell, binding with the receptors and producing any type of cellular responses. That's called a chemical messengers. Now let's discuss about a messenger binding. How these messengers are binding on the binding sites of receptors, that we'll discuss in this one. Usually these messengers are binding with the binding receptors. You can see here receptors. This is a binding site. You can see here this is a binding site. On the binding side, there is three different type of bonding forces are working where chemical messengers are binding with this different type of bonding forces. First is ionic bond. You can see here ionic bonding between the ions. This is called a ionic bonds. Next, hydrogen bond, and third is van der Waals bonds. So each and every chemical messengers, they are binding with a receptors due to these three types of bonding forces. If they are properly binding along with these three bonding forces, then only they can activate a messengers or the binding sites. They can activate a receptors only if they are binding properly along with these three bonding forces. Then only they can bind it with the properly with the receptors and they can activate it properly. So these three major bonding forces are important. First is ionic bonding, hydrogen bonding, and van der Waals bondings. You can see here messengers. This is a binding site. Three different type of bonding forces are there. You can see here van der Waals interactions, ionic bonding interaction, and hydrogen bonding interaction. If these three interactions are properly binding with the messengers, then only they can induce a binding fit. Next, under a bonding forces, we'll discuss about a induced fit. Induced fit means when the messengers are binding properly with these three type of bonding forces, in that case they can slightly change or receptors binding site slightly change their shape for inducing a fit of messenger on a binding site. Once again I'm repeating there, on a messenger binding site, when the messengers are binding properly with these two types of bonding forces, it will induce a induced fit. Due to this induced fit, this binding site of receptors slightly changing their shape for binding of a messengers to a binding site. That's called a induced fit. Next is, now here they are showing a overall process of receptor and messenger interactions. When the messengers are binding on a binding site of receptors, this receptors binding site slightly changes their shape due to the induced fit. So the messengers can easily bind with the receptors and they can pass their signals inside a cell. And after passing their signals, they can easily release from a receptor binding site. So there is three major properties are there. First, a interaction between a messengers and a receptors binding site should be strong enough to hold a messenger sufficiently longer time so that they can easily transduce their signal. First property is there, the messenger should be bind with receptor for a longer time or sufficient longer time so that they can pass their signals to a receptor or to a cell. Next, their interaction between a messenger and the receptor should be weak enough also so that after passing their signal they can allow to depart a messenger. They can allow to depart a messenger. So means the interaction should be in a fine balance. So these three are a major property of receptor messenger interactions. Last point is that designing molecule with stronger binding interactions result in drugs that block the binding site. If messengers are binding with the receptors, if the messengers are binding with the receptors and they are not properly binding with three bonding forces, then they can block a receptors. That's called a antagonist. That will discuss in the next slides. Now let's discuss about a receptor superfamilies. There is a common four different type of superfamilies are there. First, ion channel receptors, and their action or the response time is in milliseconds. Next, G protein coupled receptors and kinase link receptors. Their response timing is in seconds. And last is intracellular receptors. Their response timing in minutes. So let's discuss one by one about all these receptors. First is ion channel receptors. You can see here in a figure, they are showing a cell membrane or a plasma membrane. So inside a plasma membrane, different type of receptor proteins are embedded here. You can see here on a plasma membrane, different type of receptors proteins are embedded there. So these receptors proteins, they are called a ion channels. You can see here different type of ion channels are present here. So you can see here this type of channels are present inside a cell membrane, or this is also called as a hydrophilic tunnel. This is also called as a hydrophilic tunnel. Now here you can see, so this hydrophilic tunnel structure is showing like that. In the structure there is a gate. That gate is always, or in a inactive condition, that gate is always closed, and they are not allowing any type of ions to move inside or to exit from a channels or from a cell membrane. These ion channel receptors, these ion channels are present on a cell membrane. They having a gate. On the usual time or in a normal time, this gate are always present as a closed condition, and they are not allowing any type of ions to move inside or outside a cell. So whenever and they having a binding site for a receptors also. You can see here on the receptor there is a binding site for the messenger. So whenever the messengers are binding on a receptors, then only they will allow to open a gate and allow to enter a ions to move inside or to exit outside as per the ion channels properties. You can see here messengers are binding and they allow to open a gate. Now the ions can easily move outside or inside a cell. You can see here. As we know about the ions in our body, different type of ions are present here. So the specific ions are sodium, calcium, chloride and potassium. These four ions, they are involved in a action potentials. These four ions are involved in a action potentials. So whenever these ions concentrations are changing, whenever these ions concentrations are changing inside a cell, they are generating a, or they are producing a polarization or depolarization of a nerve membranes. So whenever the ions concentrations are changing inside a cell, they are producing a polarizations or depolarization of a nerve membrane. That is the importance of ion channels. Once again, you can see here, example is nicotinic receptors. You can see here in the nicotinic receptors, it is a 5 transmembrane receptor. There's a, in the red color you can see here, it's a binding site for a receptor. So on these binding sites, nicotinic molecules are binding and they allow to open a channels, and inside the channels they are allowed to transfer a ions. Now let's discuss about a structure of ion channel receptors. You can see here this ion channel receptors are a 4 transmembrane receptor subunits. Ion channel receptors having a 4 transmembrane receptor subunits. Why it's called a 4 transmembrane? This protein subunits are like a thread-like structure. They having a thread-like structure. You can see here on a cell membrane, this protein subunits, they have crossing this cell membrane 4 times. They are crossing a cell membrane 4 times. That's why it's called a 4 transmembrane. One end is always attached with a amine group and the other end is always attached with a carboxylic group. So that is a basic structure of ion channel receptors, and you can see here this is a proper binding site where the messengers are binding with the receptors and allowed to open a ion channels. So what is ion channels? Ion channels are a receptors which allows to enter or to exit a different type of ions inside a cell or outside a cell. That's called a ion channels. Again, you can see in this figure, this is you can see here this is a binding site on ion channels where the messengers are binding, producing a induced fit, allowed to open a ion channels and allowed to enter ions to move inside. Next is G protein coupled receptor. G protein coupled receptor is also called as a 7 transmembrane receptor, or it's also called as a serpentine receptor. So sometimes in exams they are also asking like the questions is what is the which receptor is called as a 7 transmembrane receptors, or which receptor is called as a serpentine receptors. So this G protein, this is a synonyms of G protein coupled receptors. So now let's discuss about a G protein coupled receptors. You can see here in a figure, again there is a yellow color structure, they are showing a cell membrane. This blue color structure, they are showing a receptors, and these are the binding sites. This is a binding site for the messengers. When the messengers are binding on a receptors, they are allowed to induce a fit. When they inducing a fit, this closed gate allowed to open. This closed gate inside a cell, or it's also called as a intracellular gate, which is also attached or present on a receptor. This is allowed to open. You can see here this trimeric structure. This trimeric structure is also called as a G protein subunits. This trimeric structure is having a 3 molecule or a 3 subunits. It's called a alpha, beta, gamma subunits. This trimeric structure is called a G protein subunit structure. They having a 3 subunits. It's called a alpha, beta, gamma subunits. So after binding with the messengers, this will activate or allow to open this closed gate, and now this trimeric subunits will bind with this closed gate. You can see here, and now after activation of this closed gate, this trimeric subunit will dissociate in a 2 subunits. One is beta gamma, one is alpha subunits. And later this alpha subunits will bind with a different type of enzymes, and they will produce a different type of activities. Later we'll discuss about all this. Let's discuss about a structure. They having a 7 transmembrane regions. That's why it's called a 7 transmembrane receptors or serpentine receptors. One end is attached with the amine group, other end is attached with a carboxylic group. You can see here extracellular loops which is present outside a cell, intracellular loops which is present inside a cell, and the variable intracellular loops you can see. And here you can see the G protein binding site. Why they are showing it intracellularly? As in our last figure, we saw there whenever the messengers are binding on a extracellular part of receptors, they will activate or they will open a intracellular regions where the G proteins subunits are binding. That's why they are showing a G proteins binding regions here inside a cell. So there is a different type of ligands or a chemical messengers which are binding on a G protein coupled receptors and activating the receptors. The examples are monoamines like dopamine, histamine, noradrenaline, acetylcholine. Nucleotides, lipids, hormones, glutamate, calcium ions. These are the different type of chemical messengers or the ligands which are binding on a G protein coupled receptors and activating them. Now here in this structure we can see different type of binding sites of G protein coupled receptors. You can see here in figure A. In figure A, this is a binding site. You can see the binding site inside a pocket. Inside a pocket, this is a binding site for the monoamines. Next B. In the next B, you can see a binding site on the top of transmembrane helicals present extracellularly and nearby the N-terminal chain. This is a binding site of peptide hormones. In figure C, you can see binding sites present extracellularly and nearby the N-terminal chain. Why it's called a N-terminal chains? Because this chain end part is attached with the amine group. That's why it's called a N-terminal chains. This is a binding site for a hormone. Last is glutamate. In the glutamate, the binding site is nearby N-terminal chain. So this is different binding sites for a different type of chemical messengers or a ligands on a G protein coupled receptors. Next, let's discuss about a tyrosine kinase link receptor. This is the third type of receptors, tyrosine kinase link receptor. Again, you can see in this figure, messengers are binding on a binding site, activating a closed gate. This activation of this closed gate will produce a different type of cellular responses. How they are producing a cellular response, that we'll discuss in a next slides. So first we'll discuss about a structure. This tyrosine kinase link receptor is a single transmembrane structure. You can see only one time this thread is transferring to the extracellular to the intracellular. Is called a single transmembrane structure, and terminal or the extracellular terminal is attached amine group, intracellular attached with a carboxylic group. Now in this figure, it's about a mechanism of tyrosine kinase link receptors. You can see here there is a cell membrane. On a cell membrane, two monomers are present. You can see here the pole like structure. This is two monomers. Each monomers having a 3 tyrosine kinase region of protein. You can see here in a monomers, each monomers having a 3 tyrosine kinase regions of proteins, alpha helix in a membrane, and a binding site for a molecule or for a ligand or for a messengers. Along with this, nearby the cell membrane, you can see inactivate proteins also. So whenever the signal molecules or a ligands or a chemical messengers are binding on this binding sites, these monomers will attach and they will produce a dimers. They will convert in a dimers. And after conversion in a dimers, this tyrosine kinase, they will activate and they will phosphorylate this dimer structure. They will phosphorylate with dimer structure. How they will phosphorylate? What is the meaning of phosphorylate? Transferring of one phosphate molecule from a ATP molecule and attach with the other group or proteins group. That's called a phosphorylation. What is phosphorylation? Relation of phosphate with the proteins. Relation of phosphate with the proteins. That's called a phosphorylation. So here you can see on a tyrosine kinase, this phosphate group now attaching with the proteins. This inactivate proteins will activate, and now after activation of these proteins, they will produce a different type of cellular responses. That's called a tyrosine kinase link receptors. Once again, I am explaining about this one. This tyrosine kinase link receptors having a 2 monomers, each monomers having a three tyrosine kinase regions of proteins having a binding sites where the molecules are binding. After binding of molecules, they will produce a dimers and they will cause a phosphorylation. Due to the phosphorylations, this inactivate proteins will become activate, and after activation they will produce a different type of cellular responses. That's called a tyrosine kinase link receptor. So here the example is given: epidermal growth factor receptor. This epidermal growth factor receptor is an example of tyrosine kinase receptors. In a figure we can see here on a cell membrane two monomers are present, each monomers having a red color binding site where the epidermal growth factors are going to bind, and after binding they will dimerize a structure and it will cause a phosphorylation. ATP, this protein phosphate molecule will release from a ATP and it will produce a phosphorylation and activate a proteins. That activation of proteins will produce different type of cellular responses. Next and last is intracellular receptors. As the name indicated, intracellular receptors. Intracellular means inside a cell. So these receptors are present or usually staying inside a cell. So first we'll discuss about a structure. You can see here the structure. There is no transmembrane. Why there is no transmembrane? Because this cell receptors are present inside a cell. You can see here there is a binding site. One end is attached with a carboxylic group, other end is attached with a amine group. The example of this type of receptors are steroids or steroid receptors. So most of the steroids or the hormones, they are binding through this intracellular receptors. You can see in this figure. Suppose we discuss about a steroid hormones. The steroid hormones, they can diffuse a cell membrane. Steroid hormones, they can easily diffuse a cell membrane. After diffusion, they can bind with the intracellular receptors and they can make a intracellular receptors complex. After making a complex, they will transfer to in a nucleus. They will enter in a nuclear, binding with a different region of DNA, initiating a transcription of mRNA, and later they will produce a different type of protein synthesis. That's called a intracellular receptors. What is intracellular receptors? A receptors which is present inside a cell. That's called a intracellular receptors. Examples are steroid hormones. These steroid hormones can easily diffuse a cell membrane. After diffusion, they will bind with the intracellular receptors, making a complex. This complex later can transfer into inside a nucleus, and inside a nucleus they will bind on a different elements of DNA, starting to transcript of mRNA, and later this mRNA will help to produce different type of protein synthesis. That's a major role of intracellular receptors. So like that we have discussed about all this type of receptors: ion channel receptors, G protein receptors, tyrosine kinase receptors, and intracellular receptors. So there is a summary is given in this slide. You can see here the first, ligand gated ion channels. In this ligand gated ion channels, these channels are present on a cell surface. When the messengers are binding on a channels, they will allow to open a gate and the ions can enter inside a cell. They can change a membrane potential, producing different type of cellular responses. Examples are nicotinic acetylcholine receptors, and their response timing is in a milliseconds. Next is tyrosine kinase link receptors. Whenever the messengers are binding on a receptors, it will dimerize a structures, phosphorylate a proteins, activate a proteins, producing a different type of cellular responses. Examples are insulin receptor. Third is G protein coupled receptor. When the messengers are binding on a G protein coupled receptor, they will start to produce a second messengers, that is cyclic AMP, that we'll discuss in the next slide. And after producing these second messengers, they will produce a different type of cellular responses. Examples are beta adrenergic receptor. Last is ligand activated transcription factor. This ligand activated transcription factors receptors, they are present inside a cell. After making a complex with the steroids, they will enter inside a nucleus and transcript a mRNA, protein synthesis and producing different type of cellular responses. Example are estrogen receptor. So like that, up to that, we have discussed about a, okay, just a moment. Now up to that, we have discussed about a different type of receptors, how their structure, what is the structure of the receptors, how they are binding, how they are producing their actions. Is it clear, or do you need to ask any question from there? Is it clear?
Clear.
Or do you want to ask any question? So shall we proceed for the next slide? Shall we proceed?
Yes, please.
Okay, so now let's move to the signal transduction. Now our next topic is signal transductions. As a name indicates, the signal transductions, like us, if we want to communicate with each others, we are using different type of language, or we are using any type of signs and symbols through our hand. So like that, we are communicating to each others. Same like that, in our body, our cells are also communicating to each others through passing a signals. So these signals, they are passing in a form of chemical messengers. So these signals, they are passing in a form of chemical messengers. So each and every cell has the ability to receive or to act on a signals. So this is called a signal transductions. So in this topic, we'll discuss about a signal transductions, how they are transducing their signals from one cell to other cells. So each and every cell is having the ability to receive or to act on a signals. These signals represent, or these signals having a informations. So these cells, whenever they are receiving a signals, they are analyzing a signals, or as per their signals, they are producing or changing a different type of structures or producing a protein synthesis. So these signals, they are passing in a form of chemical. So the conversion of information into a chemical changes is called a signal transduction. There is a two type of process under signal transductions. First, whenever the messengers, chemical messengers, or a ligands or neurotransmitters are binding with the extracellular receptors. This is a first procedure. Binding of chemical messenger with the receptor is called a first procedure. Second procedure, after binding a receptors, these receptors, they are changing or activating a second messenger inside a cell. They are activating a second messenger inside a cell. This is a second procedure. So there is a three specific features are there for a signal transduction. First is specificity. As we discussed in our earlier slide, for each and every molecules, they having a particular receptors. For each and every molecules, there is a specific receptors are there where they are binding. Next, affinity. Affinity, the interaction between the receptors and a chemical messengers is the affinity. There is a strong interaction or in a fine balance interaction between a receptors and a signal molecules or a messengers. Third, cooperativity in receptors ligand interaction result in large change in receptor activation with small change in ligand concentrations. Means when the molecules are binding on a receptors, this receptors having a or inducing a large changes inside a cell. That's called a cooperativity. So there is a three specific features of signal transductions: specificity, affinity, or cooperativity in structures and ligand interaction. Now, what is second messengers? Second messengers are a molecule that relay signals from receptor on a cell surface to a target molecule inside a cell. That's called a second messenger. Suppose if we discuss about a hydrophobic molecule or hydrophilic molecules, few molecules are there which are not able to transfer a cell membrane. Few molecules are there which cannot transfer a cell membrane. So these molecules can only bind on a receptors which is present on a cell surface. So after binding with the receptors, they are activating a few molecules which are present inside a cell. This molecule is called a second messengers, and later this second messengers passing that information to a inside a cell or inside a nucleus. Usually the second messengers, they are relaying a signals of hormones like ephedrine, growth factors and other hormones. So now we'll discuss about a different type of second messengers. There is a three type of messengers are there. First is hydrophobic messenger molecules. Hydrophobic, as we know about the hydrophobic, water insoluble, which hate a water. This is a water insoluble molecules like diacylglycerol, phosphatidyl inocytosols, which are membrane associated and molecules are there. So how they are activating, what is diacylglycerol, that we'll discuss later in the slides. So specifically there's a three type of secondary messengers are there. First, hydrophobic. Hydrophobic are water insoluble. If they are water insoluble, then they can't move inside a cell, because inside a cells they having a liquid property. So this molecule can't dissolve in a liquid, so they always present on a cell membrane. Examples are diacylglycerol and phosphatidyl inocytosols. Next is hydrophilic. Hydrophilic, water lovable, so they can easily soluble in a water. So as we know about our, in the cell we have a cytosol, in the cytosol we have a cytoplasm. The cytoplasm having a liquids, this liquids having a water like nature. So these hydrophilic molecules, they can easily dissolve in a water like natures. Examples are cyclic AMP, cyclic GMP, IP3 and calcium molecules that we'll discuss later in this lecture. Third is gaseous. In the gaseous, nitric oxide and carbon monoxide, hydrogen sulfide are the example. They can diffuse both cytosol as well as they can cross a cell membranes also. So there's three type of second messengers. First, hydrophobic molecules which are present on a cell membrane. Second, hydrophilic molecules which are present inside a cell. Inside a cell where? In a cytosol and cytoplasm. Third, gaseous molecule. These molecules, they can cross a cell membrane as well as they can dissolve in a cell also. So now we'll discuss about a signal transduction pathway through the activation of adenylate cyclase by GS protein. What is this GS protein? This S word is denoting a stimulatory. G stimulatory protein. So in this figure, we can see here in this figure, there is a cell membrane. On a cell surface, receptor is present. This receptor is called a G protein receptor. On a receptor, there is a binding site where the messengers are binding. Along with this, alpha, beta, gamma subunits are present. G protein is called a G protein subunits, and this subunit is present in a inactive form. Inactive form, and GGP molecule is attached with this subunits. So whenever the messengers or the molecules are binding with the receptors, receptors will activate. How they will activate? After binding with this one, this GDP molecule will replace by a GTP. This GDP molecule will replace by a GTP, and this GTP molecule will activate this subunits, and after activation, this trimeric subunit will dissociate in a two subunits. One is alpha gamma, other is alpha. So the alpha gamma later will attach with this receptor only, and the gamma later will attach, move and activate a adenylate cyclase effector molecule. This adenylate cyclase effector molecule is also present on a cell surface, and after activation of this adenylate cell cyclase, they will produce a second messenger cyclic AMP through the help of ATP molecules. Later, this cyclic AMP will work as a second messenger. This cyclic AMP will activate a inactive protein kinase A, and they will activate a protein kinase A. This protein kinase A will produce a phosphorylation. Phosphorylation means relation of protein with a phosphate molecule with a protein, and they will activate a proteins, and this protein will produce a different type of cellular responses. That's called a signal transduction pathway through the activation of adenylate cyclase by GS protein. So once again, I'm explaining this one. On a cell membrane, We can see here a receptor. Receptor is attached with a inactive form of trimeric G protein. This is a binding site where the messengers are binding. After binding with the receptors, this trimeric site will activate. And after activation, this trimeric site will dissociate in a two parts. One is gamma beta and one is alpha. Later, this alpha will move to a effector adenylyl cyclase molecule or adenylyl cyclase part. This adenylyl cyclase is an enzyme. So after binding with this adenylyl cyclase enzymes, this enzymes will help to produce a cyclic AMP molecule. This cyclic AMP molecule here will act as a second messenger. This cyclic AMP molecule later will activate a inactive protein kinase A. How they activating, how they are producing this all process, that we'll discuss later in slides. After binding with the inactive protein kinase A, they will activate this protein kinase A and they will produce a phosphorylation of a protein. Phosphorylation of protein, activation of a protein, and later they will produce any type of cellular responses. You can see in this figure, there's a trimeric structure of G protein. You can see in this figure, there's a trimeric structure of G protein. This trimeric structures usually they are present in an inactive form. So whenever the GTP is replacing this GTP, they will activate and dissociate in a two subunits. One is alpha subunit and one other is beta gamma subunits. Next structure. Now we'll discuss about a inactivate protein kinase A structure. What is the structure of protein kinase A? You can see in the next figure, there is a structure, there is a 4 subunits in a protein kinase A structure. 2 subunits are a regulatory subunits, 2 behind subunits are called a catalytic subunits. In a normal process, they are always present in an inactive form. They having regulatory subunits having a binding sites for a cyclic AMP molecule. This cyclic AMP molecules will bind on a binding sites of regulatory subunits. After binding of this molecule with the regulatory subunits, they will release a catalytic subunits. They will release these catalytic subunits. Later, these catalytic subunits will enter inside a nucleus and binding with the different elements of DNA, producing a transcription and translations and changing in a protein synthesis. That is a basic mechanism of G protein receptor. So once again, we'll discuss about this G protein in this figure. There is a G protein structure. When the signal molecules are binding, they will activate this G protein. Trimeric structure will convert in a dimeric and monomeric. This monomeric alpha subunits will bind with the adenylyl cyclase enzymes. This adenylyl cyclase enzymes help to produce a cyclic AMP. This cyclic AMP will work as a second messenger. This cyclic AMP will bind with the inactive protein kinase A. And after binding with this one, this regulatory subunits will dissociate this catalytic subunits. This catalytic subunits later will enter inside a nucleus. Inside a nucleus, they will bind with the DNA elements and producing a transcription, translation, and protein synthesis. Now next, a difference between GS versus GI. GS, S denoting a stimulatory response. I denoting a inhibitory response. So I is also like a G protein receptors. In this one, this inhibitory G protein, they will inhibit a adenylyl cyclase enzymes. They will inhibit a adenylyl cyclase enzymes. That is the only difference. So if they inhibit a adenylyl cyclase enzymes, further they can't produce a cyclic AMP molecules. They can't activate a protein kinase A enzymes, and there is no cellular responses in that case. So there is a basic difference between GS and GI molecules. Next is GQ. Next is GQ. You can see in this one, GS and GI, they both are involved in a production of cyclic AMP. GS, they are helping to produce the cyclic AMP second messengers. GI, they are inhibiting a production of cyclic AMP. And this GQ, they are coupled with a G protein receptors and producing a calcium ions. How they are producing calcium ions, that we'll discuss later in slides. You can see in this figure how these GQ molecules, they are producing a calcium ions. The structures for the G protein structures are similar. Receptors having a trimeric inactive site agonist, whenever the binding with receptors, they will activate this trimeric site, dissociate in a two parts. This alpha part will now here they will bind with a phospholipase C. In case of GQ and proteins, this alpha structure will bind with a phospholipase C. This phospholipase is a enzymes which later dissociate PIP2 molecules in a two different molecules, DAG molecules and IP3 molecules. This PIP2 is a phosphate inocytosol diphosphate, phosphoinocytosol diphosphate. So this phospholipase C enzymes will dissociate this phosphoinocytosol diphosphate in a diacylglycerol and IP3 molecules. This diacylglycerol is a hydrophobic in nature. This diacylglycerol is a hydrophobic in nature. They can't enter inside a cell, so they always present on a cell membrane. So later this DAG molecule will bind with the protein kinase C enzymes. This protein kinase C enzymes, after binding with the DAG, along with calcium ions, they will activate and producing a cellular responses. Next, about a inocytosol triphosphate IP3 molecules, they are hydrophilic in nature, means they can dissolve inside a cell also, they can move inside a cell also. Later this IP3 molecules will bind with a endoplasmic reticulum, and this endoplasmic reticulum is having a calcium ions. So this calcium ions, after binding with the IP3 molecule, will release and enter inside a cell. Inside a cells, this calcium will bind with a protein inactive protein forms, and they will activate the proteins and producing any type of cellular responses. So this is a phospholipase C enzymes activity. You can see in this figure, this is a PIP2 molecule. In presence of phospholipase C enzymes, it will dissociate or it will convert in a two parts. One is GAG molecule and other is IP3 molecule. This GAG molecule is a hydrophobic in nature, so that's why they always present on a cell membrane. IP3 molecule is a hydrophilic in nature, they always present inside a cell. IP3 molecules helps to release a calcium ions from a stored condition of inside of endoplasmic reticulums. And this DAG molecule, along with the calcium ions, they will activate a protein kinase enzymes and producing different type of cellular responses. Last is this calciums as second messengers. These calciums are releasing, as we discussed in our earlier slides. This IP3 molecules will bind with the endoplasmic reticulums. This endoplasmic reticulums having a storage of calcium ions. They are helping to release these calcium ions inside a cytosol. Later this calcium ions will bind with the protein kinase enzymes and they will activate this protein kinase. So this is a calcium as a second messengers. So this is our next second part of the signal transduction pathways. So do you have any queries or any questions in the receptors and signal transductions? Do you have? If you have any queries, you can ask me. Is it clear? So now before starting our next lecture, shall we take a 15 minutes gap? Okay, so right now is a right now timing is 4 o'clock, so we'll meet around at 4:20. Okay?
Okay.
Okay, then see you. Bye.
Bye. Thank you.
Welcome.